The Biosynthesis of Vancomycin. Mycoletopsis orientalis is a microorganism or microbe usually found in soil. It is a unicellular filamentous bacterium that grows into a branching network of filaments. A Mycoletopsis has evolved to produce secondary or specialized metabolites that interfere with the growth of competing bacteria. One of these metabolites is vancomycin, a compound that has earned an important role in modern medicine as it is used as a last resort antibiotic against multi-drug resistant pathogens. Vancomycin prevents pathogens from stabilizing their cell walls by interfering with key cell wall forming reactions. The cell walls of these doomed pathogens cannot be properly formed and the pathogens explode. Vancomycin was discovered in the 1950s, and its structure and mode of action are well studied. We will focus first on its natural production, or biosynthesis. Part A, biosynthetic gene cluster. There are many processes taking place in a bacterial cell, such as growth, development, and reproduction. These are necessary for survival and belong to so-called primary metabolism. Additional processes are only activated under certain conditions and comprise so-called secondary or specialized metabolism. The products of secondary metabolism include antibiotics, pigments, or scents. Secondary metabolites are produced by microbes like a Mycoletopsis orientalis living in crowded habitats, and it is likely that a Mycoletopsis orientalis produces vancomycin as a defense against competing bacteria. The blueprint for vancomycin biosynthesis is hidden in the genome of the bacterium. The genes that encode vancomycin are located in close proximity to each other, forming a biosynthetic gene cluster, called a BGC for short. The biosynthesis of vancomycin is quite complex, involving elaborate production machinery. All of the necessary steps for vancomycin production can be traced back to enzymes encoded in this single BGC. Part B, backbone assembly. In order to produce vancomycin, the necessary building blocks, 7 amino acids, need to be obtained or produced. 2 of the amino acids occur naturally in the cell. The others, called non-proteogenic amino acids, need to be made. The precursors of these non-proteogenic amino acids are derived from primary metabolism, and the enzymes required to synthesize them are also encoded in the BGC. The second step is to build a chain out of the 7 amino acids by connecting them, end to end, through peptide bonds into a heptapeptide backbone. The assembly line for the backbone is built from 3 non-ribosomal peptide synthetases, NRPSs for short, that are themselves connected via their docking domains. NRPSs are large multimodular enzymes that build up the backbone of vancomycin step by step. These 3 NRPSs consist of 7 modules, and each module consists of different domains, and each domain is responsible for activating and connecting one amino acid. This works as follows. The A domains are responsible for recognizing one specific amino acid. They catalyze a 2-step reaction. First, an amino acid is activated and then transferred to the PCP domain. PCP domains are used to shuttle amino acids and peptide intermediates to the pocket of the next domain, the C domain. C domains catalyze amide bond formation between 2 loaded amino acids. They have 2 pockets, one for the amino acid or peptide upstream and one for the amino acid downstream. C domains transfer the amino acid or peptide from an upstream PCP domain to the amino group of the amino acid that has been previously located onto a downstream PCP domain. Some modules include an E domain, which catalyzes the conversion of L amino acids to D amino acids. Let's have a closer look. The last module contains the TE domain. This last domain breaks the thioester bond between the heptapeptide and the PCP domain, releasing the peptide into the cytosol. The entire backbone of vancomycin has now been assembled. But before the TE domain releases the peptide, a special process must be completed during the biosynthesis of vancomycin. Part C, modifications during backbone assembly. Some of the modifications of the amino acids occur during backbone assembly. One modification, called halogenation, is catalyzed by the enzyme halogenase. This enzyme adds a chloride to the two BHT amino acids as soon as they are incorporated into the peptide at the 2nd and 6th module. Another modification, the cyclization of aromatic amino acid residues of the non-proteogenic amino acids, is performed by enzymes called P450 monooxygenases. Oxidative crosslinks are formed by 3 of these enzymes, acting in a specific order at the heptapeptide, each one being responsible for a specific oxidative link between 2 amino acids. They are recruited by the X domain, which only occurs in the last module. OxyB catalyzes the first link between the 4th and the 6th amino acid. OxyA catalyzes the second link between the 2nd and the 4th amino acid. Finally, OxyC links the 5th and the 7th amino acid. Now that the heptapeptide has 3 crosslinks and the 3-dimensional form it needs to bind to its target, it is released from the assembly line. Usually the machinery does not wait until one peptide is ready, but the synthesis of multiple backbones happens simultaneously. Part D, final modifications. After release of the peptide, 2 more modifications are needed. First, 2 glycosyl transferases ensure the attachment of sugar to the peptide. The first sugar is attached to the central amino acid, and the second sugar is attached to the first one. The second modification is catalyzed by a methyltransferase that attaches a methyl group to the first amino acid. Now, the biosynthesis of vancomycin is complete. Vancomycin is transported out of the cell and secreted into the environment, where it can kill competing bacteria by binding to a compound called lipid 2, a key building block in bacterial cell wall construction. By binding to lipid 2, the enzymes that normally link lipid 2 moieties together and stabilize the cell wall are prevented from doing this. As a result, the cell wall cannot be properly formed and the cells explode. This particular method of fighting bacteria has been adopted by modern medicine in order to fight multi-drug resistant pathogens when other antibiotics won't work. For this reason, vancomycin is referred to as an antibiotic of last resort, and it has saved countless lives.